transfusion practices, ensuring safety and enhancing patient outcomes. Here are some key areas and consideration in this field. First, data management and integration through electronic health records and blood management system. The second, blood safety and testing, pathogen reduction technology, research into new methods for pathogen inactivation to reduce the risk of transfusion-transmitted infection, and molecular testing, inventory management, predictive analytics, and real-time monitoring, collaboration and networking, blood bank collaboratives, regional or national networks facilitate the sharing of best practices, research findings, and data analytics to improve outcomes, and also interdisciplinary research, collaboration between hematologists, transfusion medicine specialists, and data scientists to explore innovative solutions. Education and training through simulation training and also continuing education. And of course, patient-centric approaches and regulatory compliance and quality control, where standardization of procedures and quality improvement initiatives will be the most important thing. Effective research sharing in blood bank technology is crucial for advancing the field. It is not only enhances safety and efficiency, but also promotes collaboration among healthcare professionals, ultimately benefiting patient care. By leveraging data, technology, and collective knowledge, blood banks can continue to innovate and improve their services. Thank you, and hope you enjoy the meeting, the webinar today, and of course, collaborating, sharing experiences between us.
Thank you. Before moving on to our research sharing, let's have some pictures. Turn your camera on and give your best smile. Okay, now for first slide. One, two, three, smile. Okay, next. Second slide. One, two, three, smile. Okay, next. Third slide. One, two, three, smile. Next. Okay. One, two, three, smile. Next. One, two, three, smile. Next. One, two, three, smile. Okay, last one. One, two, three, smile. Okay. Thanks. Okay. Dear participants, we have come to our exciting session, which is our research sharing. This session will be moderated by Mr. Wisnu Budiwaluyo. He serves public relations and international affairs at Stikes Gunabangsa Yogyakarta. Complete his bachelor's in international relations. Mr. Wisnu Budiwaluyo is passionate about policy and development studies with his master's in development and sustainability. He is now focusing on environmental and public health. Mr. Wisnu is also a lecturer on our campus, teaching some courses including health policy, public health, health ethics and law, and many more. Without further ado, over to you, Mr. Wisnu.
All right, can you hear me? Good, all right. All right, everyone. Assalamualaikum warahmatullahi wabarakatuh. Okay, good morning. Selamat pagi, welcome to our webinar. And our distinguished guest, Dr. Isma from the University Technology Mara. Thank you so much for joining us today. And we also have our second speaker, Mrs. Gravinda. Thank you so much. And our third speaker, Dr. Fitri. Thank you so much for joining us. And this is the moment that we are waiting for, because we will hear the research sharing from our speakers, from their expertise, from their experiences. So we will learn a lot from them. So the first speaker, of course, as you can see that today we have Dr. Isma. So Dr. Isma is from University Technology Mara, Malaysia. Okay, let's have some CV of Dr. Isma. Associate Professor Dr. Wan Ismahanisa Ismail is the head of Center Faculty of Health Sciences, UITM, Campus Bertam, Penang, Malaysia. So it's such an honor for us to have you today, Dr. Isma. And Dr. Isma earned her diploma in science and also Bachelor of Sciences in Medical Laboratory Technology with honors. And also she completed her master in transfusion science and also PhD in pharmacoepidemiology. So her research fields in thalassemia, transfusion, and she is also senior lecturer, researcher, and adjunct professor and research fellow at Universitas Airlangga, Surabaya, Indonesia. So this is our pleasure, our honor to have you, Dr. Isma. So please stay tuned, and then you can listen to her research sharing, and you can prepare your questions, and then our committee will provide the chat box, the link for the questions. So enjoy the session with Dr. Isma. Over to you, Dr. Isma.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much for your kenalan ya. Okay, I'm really glad, okay, to here together with you all at the Stikes Gunabangsa. Together with me, all my students from the Medical Laboratory Technologies. That previously you are introduce myself. Okay, I'm not longer the head of center of the faculty. It's already been 5 years. I'm held the position of the head of center. Just a few months, I'm already free for the positions, so free to fly everywhere. So, in other way, I'm also as a supervisors of all the PhD student, all the 10 PhD student, and also the master students, and I also the advisors of the Thalassemia Society in Kedah and Sabah also. So can be open. Oh, okay, okay then. Then we begin start of the blood transfusion management for thalassemia patients. But before, can we open the video first? Then the students also knows about the thalassemia more detailed compared to this our This video is about of my patients, okay? 3 of them are kids with the thalassemia. Okay, I'm together with the last one's children, Aulia. So they already been within the 4 months start transfused from the age of the 4 months. So sampai sekarang umurnya 6 tahun. So kita dah rapat dan kita rasa suffernya. So can we go open it, the video first? Yeah. You already open the video? Video. Can they open the video? Boleh dia. Can they open the video? Okay, bisa. Okay. I think it's some have a technical problem. I think it's okay. We start with the presentations first. Okay. So in the way of the blood transfusions management for thalassemia patients, okay. So actually you must know about the thalassemia first, okay, the need of the thalassemia patients, what their needs, okay. In Malaysia, okay, normally the thalassemia patient is all over scattered in Malaysia, okay. We have the 14 state in Malaysia, we have the 14 organizations, the Society of the Thalassemia Patients, to handle of the thalassemia patient in Malaysia. So the numbers of the thalassemia patient in Malaysia is around 11,000, okay, 11,000 with total with the beta thalassemia major. Okay, so you must know, okay, all other students must know what is thalassemia. Is that the thalassemia can be infectious, can be transmitted, okay. So did you know, kalau you tahu tak, okay, thalassemia ni berjangkit atau tidak? Okay, selalunya orang kata, semalam kita tanya the students that you know the thalassemia ni berjangkit ke atau tidak? So dia katanya transmit, infectious ya. So tidak. So the thalassemia is called the genetic disorder. It's totally genetic disorder. It can be transmitted. It can be via if your coughing is, or if you are eating in the same place. No, it's not being happens. Okay, it being happen if you are married with the carrier with carrier. Then of course you akan ada anak yang thalassemia. So it's very clear cut. If you are involved with genetic, it's very clear. Perkahwinan, the married one. Okay, so the thala is come from the sea. Okay, that's why they call it thala. Mia is about the blood disorder. Okay, when it's involved in blood disorder, okay, so But the pity of the thalassemia patient, okay, for the age of every month, okay, there are needs of the transfusions. That's the mandatory of you guys, okay, of the blood bank, okay. Tabung darah ini adalah fungsi yang paling penting untuk pesakit thalassemia. Of course, if you are not longer in the world, okay, in Indonesia, what happened for the thalassemia patients? Can we buy a blood in the Indomaret? No. Okay, I dah pergi Indomaret, it's everywhere in Indonesia. So, you cannot buy a blood dekat di mana-mana. If must be prepared by the tabung darah. Okay, so you are a very important person, okay, in the situation of the thalassemia patients that needs of the blood. Okay? It's every month, it's every single of 2 weeks they need of the blood pack. Okay, so then after that, the screening of the thalassemia, okay, that is very important. Okay, if you want to prevent of the thalassemia, okay, you must do a screening. But before you do the screening, you have to look back of the viruses infection in your body. Okay, so then if you have the hepatitis B or not, hepatitis C, HIV, normally in the patient of the thalassemia, okay, if you are in the 20 years, okay, they are start from the cradle until to the grave. The start from the age of the 4 months is very earlier of the stage age, okay, until dia meninggal dunia, until death, okay. They are carry of the genetic of the thalassemia and are depend on the transfusions in the long life. So, if these are the way yang you need to prevent to get the thalassemia. Yes, you are healthy now. You are carrier one. But your generation afterwards, your anak. Okay. So the transfusions is in thalassemia. Okay. This is very kata major components. Okay. The major treatment after you have the thalassemia disease. Okay, the combinations of the transfusion and ion chelation therapy, tak boleh ditinggalkan oleh pesakit thalassemia. These are very important. But in the way of the before you do the transfusions, okay, you must do the ABO blood grouping. This is very basic things yang kita buat dalam lab. Okay, the basic component, the basic ABO blood grouping of the patient as well. Okay, RHD group. And then the major antibody you need to identify in the blood of the patient as well. And also the donor, because we must do the matching. If you are not matching of the blood component between the donor and recipient, what happened to the patients? Okay, for example, in Malaysia, okay, if a certain hospital in Malaysia are not doing the allo antibody screening, for it's called the antibody identifications. So it happened in Indonesia together. You just stop until the antibody screening. But what happened if the so many antibody are forming after the blood transfusions? That's why the patient until 20 years, umur dia 20 tahun, there has no blood component dia boleh terima. Because the rejections are very high. Sampai the grades 4. Bermaksud dia harus signing of the when they are tanggung sendiri risiko kematian selepas menerima darah. Because of these matters. Okay. So that's the yang issue, the complication will happen towards the thalassemia patients. But the goal of the transfusions, okay, the goal of the transfusions is to ensure the sufficient, to ensure the blood component are received by the thalassemia is very good. Okay, it's very, orang kata, the very best things to give the thalassemia patients. Because, for example, for my patient in Malaysia, okay, they need to waiting list. For example, for 1 month, contohnya the needs of the 5 is every month, for every month. But they need to long wait for until 2 weeks. But during that time, the HB level dah sampai 5. Dia tak boleh buat apa, dia just do that reagent, baring dan tak boleh buat apa dan waiting the blood. Because of this, kita tak jumpa yang the matching of the blood component. So, achievement to appropriate the HB level, okay, to minimize effects. What you are considered when you are give the blood to the patient, you must consider the effect, the adverse effect towards the thalassemia patients. Okay, normally the complications that happens, okay, it can be the sudden deaths. If there are, during the transfuse, they get the transfuse, the reaction very high. For example, that patient they have so many antibodies in the blood. Okay, they receive the blood. So there can be the sudden death. Sebab tu lah they have to the consent form, to the signing. If there is anything happen to you, it's not the hospital blame. It's happen to your decisions. That's the things yang kita tak boleh nak prevent. Okay, because of the very expensive to doing the antibody identification test. Okay, that's why we need to prevent before we going to happen of the thalassemia. So the regimen of the transfusions, okay, you need to consider optimal time to start the transfusion. Normally we start the transfusion when the HB level 9, 8. So we start the transfusions. But in Malaysia, normally we cannot until we then needs to wait until 5. Okay, they are suffers then. During that time, the oxygen, the level of the body are very low. That's why they cannot doing anything. Because they need to be bedridden. Okay, they cannot take care of their children because they are very weak. They are very fragile, cannot do anything else. Okay, so there's only way sleep and then do the bedridden from morning till night. Sebab dia kalau dia bergerak atau melakukan pekerjaan, dia akan menjadi lebih lemah dan boleh menyebabkan kematian. So how much blood to be transfused, it depend on the situations. It depend on kadang-kadang adanya terima 1 bulan 2 bag. It depend on the severity of the body. Okay, so the transfusion efficiency sama ada HB dia meningkat atau tidak, atau dia masih memerlukan darah. That's the efficiency. Kadang-kadang it's not efficiency of the after the transfuse. They still have the weak, have the pallor, have the anemic, there's no energy. So the very important issue afterwards, the adverse reaction. In Malaysia, we are doing the for blood transfusions happen in the hospital. In each hospital, we have the daycare ward. So the thalassemia patient, sorry, will coming to the hospital, okay, to do the blood transfusions, okay. So we monitor them after the 1 hour to 2 hours, okay. So if anything happen during that time, okay, we have the emergency, we can do the emergency to save their life. But during the transfusion happen, tiba-tiba the reaction happen, the shock, dia macam apa, tiba-tibanya dia breathe, okay, kadang-kadang dia short of breath. So we need to stop the immediately of the blood transfuse because it happen the adverse reactions. Okay, kita tak boleh nak kata, oh sayangnya darah, mahal ini darah. Darahnya di Indonesia dibeli ya, kalau di Malaysia free. Oh, okay, ditanggung sepenuhnya oleh government. So jadinya di Indonesia harus bayar 500,000. So jadinya, mak dia kata, jangan. Sayang ini darah 500. Dia pula perlukan 2 bag. 1 juta itu. So jadinya, so we need to stop immediately. That's why kita dipantau oleh nurse during the daycare. Have to monitor the patient of the thalassemia. Because the complications is macam satu kakinya di kematian. So that's the things yang mereka lalui setiap bulan when they go for the transfusions. So that's the begin of the transfusions that I mentioned that before. Bila dia dah rasa lemah sangat, okay, dia terpaksa ke hospital atau doktor dia akan call doktor. Dia akan call nurse atau MLT di hospital. Okay, ada tak darah saya? Dia kata, lepas tu nurse kata tak ada yang compatible lagi. So you datang hari tu, mula-mula dia datang untuk check blood dia. So and check antibody dia. Lepas tu the doctor say, nurse say, tak ada yang compatible lagi puan. Mungkin minggu depan kot. So they waiting. Pada waktu itu HB level sampai dua dia menunggu 6, 5. So then, sorry okay, for the technical problem. Okay, so then, if the character of the thalassemia patient memang kita kenal, the features dia, dia mempunyai features yang lebar. Okay, mukanya, jawnya lebar. Okay, sebab apa? Because of what? Because in every month, okay, the blood will be lysed within 2 weeks. Okay, within 2 weeks blood will be lysed. Okay, so then the bone, each of their bones ni try to produce the blood. So that's why dia punya bloods, dia punya jaw makin lebar, dia punya tulang is compact. Okay, that's why of the character of the thalassemia ni we can see the faces. Kita boleh kenal. Okay, so so what the red cell component? Okay, we have so many kinds of the red cell component. We have the packed cell, we have the red cell less than 7 days, we have the liquid depleted cell, we have the Buffy coats, we have wash red blood cell. So normally we give the patient liquid depleted RBC and also liquid filtered RBC. It depends on the patient status. Normally, patient with thalassemia, they are not only the carry of the thalassemia, because they have so many kind of the complication afterwards. Okay, for example, they have the problem with the kidney, they have a problem with the liver failure, they have the impact of the viruses. Okay, they have the problem of the cancerous. So they have so many kind of the complication after the 20 years they are get the thalassemia. So that's why we have to filter, remove the WBC to get their immunized, to get their low of the reactions. Okay, kalau banyak sangat WBC, dia akan menyebabkan penghasilan antibody itu akan menyebabkan dia detect of the reactions. So dia akan menyebabkan lebih tinggi reaction yang berlaku. So in the blood bank peoples ni, you need to be cater of these patients to aware if that the reduce dia punya minimal of the adverse reaction. Okay, penting kan you all ni kan? So it's very good. Okay, banyak orang kata pahalanya, barakahnya kalau you buat cara yang betul. Kalau you buat cara yang salah, dosanya lebih banyak. So masuk ke syurga ataunya ke neraka itu. So the selection of blood transfusions or presence of the specific electrolyte. These are the very happenings everywhere because of the safe budget. we are talking about the money expenses of doing the allo antibody identifications, because we have 12 panels of the antibody idents. Okay, in the hospital, everywhere in the hospital, even though in Malaysia, only the certain hospital are doing the antibody identifications. That's the things yang kita tak nak dan kita nak buat. Okay, because the antibodies happen in every month is developed, the new antibody. If you are not doing the antibody, allo antibody screenings or phenotyping, so the new antibody forming dalam badan darah thalassemia patient. Dan akhirnya, dia tiada darah yang dia boleh terima. Itu yang terpaksanya sign consent form tu. That's why yang kita rasa macam alamak, kita rasa bersalah. That's the point things yang kita tak boleh nak buat. It's beyond our control, because the government says, okay, this is not the budget have, because you are increase the number of the thalassemia patient, so you don't have the enough budget to do the antibody identifications. But in the way, this is very dangerous to the thalassemia patient. So, assessing the effective of the blood. Okay, normally, okay, after the 1 week of the blood component received, okay, we need to assess either the patients are in good condition. For example, if the spleen are become enlarged, okay, there is not a HB level in the good levels, okay. So normally of the thalassemia patient, they are not have the spleen anymore. They tak ada dah spleen. So because the spleen already been removed, okay, because the complication during the so many years doing the transfusions, this not only 1 year, 2 years, but the long life. Umurnya 25 tahun. 25 tahun lah dia dah transfuse. 25 tahun kali dengan 12, berapa dah? Kali pula dengan 2 setiap bulan. So how many bags they already received? So much. Okay, reason for increase the transfusion requirements, okay, because of the developments of the allo antibody. Okay, these are the way. So requirement kita lebih ketat bilanya adanya allo antibody yang berlaku dalam badan patient yang terlalu banyak. Tiba-tiba kita tengok the reaction reject. So sebab kita tak buat antibody adent. So jadi what happened to that patient of components tu? Kita memberi tanpa kita melihat, very dangerous. Okay, so menyebabkan dia akan ada quality of blood dia tidak bagus, RHB dia pun tidak bagus, menyebabkan boleh bleeding and then disruption. Okay, during dia takes the medications. Okay, they are not successful. Okay, if even though they are get the medication afterwards. So after that, okay, the adverse reactions, okay. These are normally yang we look seriously after the patient receive the blood bag. Okay, the adverse reactions, okay, because they are going to sudden deaths. Okay, pernah saya punya patient umurnya 21 tahun, baru jumpa minggu lepas katanya. Kita jumpa kan ada satu kenduri, tetapi dia bercakap ada doktor minggu depan nak transfuse blood ni, saya kata. So kita tak sangka when the age of the 21, okay, they one of the committee members in the thalassemia NGO. A very active person. But it's not kata nasibnya, akhirnya dia meninggal dunia selepas 30 minit dia have so many complications kan. By right, no component yang competitive as the reaction happen, dia meninggal dunia. So this is very kita need to look either, okay, biasanya kita akan tengok hepatitis B, hepatitis C, HIV. The now in very technology, okay, kita can preventing of the HB/HC dengan HIV, but the complication of the series of other ways tu yang akan berlaku kepada thalassemia patient. So kita kena, itu yang saya dah cakap tadi tu. We need to stop immediately. Okay, we not if dia nampak je rash, nampak dia macam dia tiba-tiba panas, so nampak dia tiba-tiba dia macam dah sesak nafas, okay, so then we need to stop immediately. Okay, sebab tu patient of the thalassemia patients tu kena supervise very well. Kita tak boleh tak tengok dia, kita tengok carefully, terutamanya melibatkan anak-anak, melibatkan budak-budak kecil yang kita tak tahu dia punya aktif tu, tiba-tiba dia tak sedarkan diri. Dia bawa ni macam darah akan diikat di tiang tu. So ada roda kan, dia akan jalan sana, jalan sini. Sebab dia boring kan, within 1 hour untuk blood bag transfuse. So dia jalan sana, jalan sini, jalan sana. Tiba-tiba je dia pengsan. Sebab dia tak terfaham. Because the children is not aware about their conditions body compared to the adults one. Okay, so then we need to stop. Okay, so these are the way the guideline is the international, it's not the national guideline, it's international guideline. I'm also the one of the community member for the International Federation of the Thalassemia. In the way in the Cyprus, we also involved with the Turkey Thalassemic NGO. In the Turkey, kita involved with thalassemia patient in dekat Turkey. Saya sebagai advisor juga di sana. So we involved with them. Kita nampak perbezaan Cyprus dengan di Malaysia atau Indonesia. Okay, they are very the policy makers. Okay, this very important issue is policy makers. Okay, they need to do the screening that before the Cyprus. Okay, there are 90% of the thalassemia patients. Now it's less than 5% with the thalassemia because they enforce before married must do the genetic. It's not the only thalassemia. They go for the genetic screening. So they are now they started for the Arab Saudi. Okay, these are the recommendation of the patients. Okay, we need to look the grade C, of course, the major antibody. So we need to do the screening for the viral infections. Okay, so then if that the patient is HB level less than 7, okay, then we look the volume of the blood pack. Okay, either the needs of the 1 or 2. Okay, but interval transfusion usually 2 to 4 weeks. Tetapi kadang-kadang bulan seterusnya pun tak dapat darah lagi. Okay, because of the limited of the blood. Okay. Okay, afterwards, after the patient receive the blood back, apa dia nak buat? Okay, the needs of the iron loading. If you get the blood, you akan lebih iron, zat besi dalam badan. If you lebih zat besi dalam badan, dia akan berkumpul di semua organ. Dia akan berkumpul di jantung, dia akan berkumpul di liver, dia akan berkumpul di kidney. Sebab itulah pasien kita ramai yang lakukan dialisis. Sebab kidney dia dah tak function. Sebab terlalu banyak iron. Liver pula menjadi ada cancerous. Jantung pula terlalu membengkak disebabkan iron berkumpul di jantung. Dan akhirnya dia meninggal bukan sebabkan thalassemia, tetapi menyebabkan myocardial infarction. That's the way yang kalau kita buat transfuse, kita kena removekan dia. So, these are the scattered of the thalassemia patient in Malaysia. Okay, these are registered. I think it's during my time studies PhD, 2017 and 2014, back then. Dulu waktu itu numbersnya 4,500 saja. Now it's almost 6 years I finished my PhD. The increasing 1 year is 1,000 is every year. Now it almost 11,000. It same happens in Indonesia. During back then 10,500. Sekarang dah hampir 18,000. Bila saya tahu darahnya kena beli di sini, langsung saya takut tu. So then di Sabah adalah yang paling besar jumlahnya. So blood supply that slide before. Yeah, this is the way. Sabah is the state yang paling tinggi thalassemianya. Okay, hampir 4,000 thalassemia patient di Sabah. Sebab tu now I'm going involved with the thalassemia di Sabah untuk do the improve the quality of life the patient as well. So number supply tu 58,000 setiap tahun. So So darahnya tak ada. Kita pun tak derma-derma lagi ni, kan? So what happened to patient? Okay, then. So these are features of the thalassaemic bloods, okay. Bila dia berubah bentuk kepada pencil, okay, shapes atau berubah bentuknya, menyebabkan dia tak boleh nak pass through the bloodstream. Bila tak boleh pass through the bloodstream, menyebabkan dia lyse destruction. Bila ada lyse destruction, menyebabkan organ-organ dalam badan ni tak dapat oksigen yang cukup. So that's why they akan become anemic. So they are called the thalassemia major. Okay. Normally thalassemia major is occur when you are stage of the 4 months. Biasanya akan detect waktu 4 bulan bermulalah episod anak itu demam berulang kali. Setiap minggunya demam, lepas tu kulitnya makin kuning, tiba-tiba matanya menjadi kuning, perut dia membesar. So because we detect there are less of the HB level, okay, menyebabkan oksigen ni tak cukup dalam badan, menyebabkan dia sentiasa demam. So that doctors need to screen the blood. Okay, we are doing the testing. So kita nampak dia punya spleen membekak. So the faces nampak berubah. So kita tahu dia adalah thalassaemic. But we do the confirm. But the age from the 4 months dah start dah. So that's why dia kata from the cradle until to the grave. Okay. So then you lose the features of the thalassaemic. So dah nampak dah jaw dia, gigi dia, muka dia, bentuk muka dia memang sama je. So this condition of the thalassaemic patients. So then so these are the way of the complication yang akan berlaku dekat thalassemia patient. Tengok betapa banyaknya berlakunya complication in every each of the body itu akan ada complication. Terutama yang paling besar adalah hormon yang tidak stabil. Testosteronnya, estrogennya, pituitary glandnya, okay, abnormal. Okay, menyebabkan dia terpaksa mengambil hormon, mengambil pil hormon, mengambil macam-macam ubatan untuk menyebabkan dia sihat. Okay. So, we go further for the lab diagnosis. Okay, these are the way yang do the, when you want to detect the thalassemia. Okay, first you have to do the FBC. Kalau di Malaysia, okay, at the age of the 16 years old, we do the screening at school. All the school student, umurnya 6 bulan, 16 tahun, akan melakukan screening thalassemia. So, they have a cards. Okay, untuk menunjukkan dia ada atau tidak thalassemia. So after kita melakukan FBC, kita tengok dia punya MCV low, MCH low, MCHC low, MCVD low, kita buat the next step, iaitu kita buat peripheral blood film dan HB electrophoresis. Next. So kita buat dengan iaitu HB electrophoresis, kita melihat dia punya HbA2, HbF dia, kita dah dia what type of the thalassemia. Tapi in the way of the thalassemia, kita tak ada alpha major. Alpha major ada, tetapi they call the neonatal of the thalassaemic. So menyebabkan dia terus meninggal dunia selepas sahaja dia dilahirkan. Okay, sebab tu tidak ada beta alpha thalassemia major yang hidup. Okay, dia hanya ada minimal minor of the alpha. Okay, so kita is the low serum ferritin menunjukkan juga indicator of the thalassaemic patient. Okay, selepas itu kita juga buat FBC, kita buat H inclusion, kita buat clear hotels, kita nak tengok what kind of the intermediate ke, kita nak tengok major ke, kita nak tengok H firm diagnostic that patient have the thalassemia. Okay. So when the laboratory findings, okay, shows of the beta thalassemia major, okay, will start at the age of 4. Okay, beta alpha, beta, the absence of beta menyebabkan HB dia low, hematocrit dia low, MCV dia low, MCH dia low, dan MCHC dia low. So all these indicators shows that the beta thalassemia major yang needs of the blood component start from the age of 4 months. Takkan ada beta thalassemia major start umurnya 10 tahun. Tak. Dia akan bermula sejak dia lahir, selepas selalunya menunjukkan pada umur 4 bulan. Bila mana spleen dia telah membengkak. Sebenarnya spleen dia sedang membengkak. Tetapi bila spleennya dah terus membengkak, perutnya dah semakin melebar, menyebabkan dia ada complication. Menyebabkan dia very sensitive to infections. Itu menyebabkan dia demam. Itu yang kerap kali demam. So kulitnya menjadi kuning terus. Matanya menjadi kuning. So that kind of the shows of the symptom. Okay, then the prevention. Okay, the only way of the prevention, all the students here, okay, you must do the awareness first. Okay, ada ya cerita saya nak suka cerita. Patient saya, 2 tahun lepas. Okay, dia mesej saya. Doktor boleh tak tengok saya? Mesej kat Facebook. Doktor boleh tak tengok saya punya lab diagnosis saya? Saya nak kahwin lagi 2 bulan ni. Nak 2 bulan baru kau bagi kenapa? Aku cakap. Dia nak tunjuk, doktor saya nak anak ni, nak anak. Dia patient. Dia beta thalassemia major. Saya nak berkahwin. Lepas tu dah tunjuk dah kad ni, jemputan ni lah jemput. Tapi dia suruh bagi lab diagnosis isteri dia. Dia baru terfikir nak buat. So, yang tidak bernasib baiknya, dia pembawa juga. Bakal isterinya pembawa. So, memang confirm lah tak boleh nak ada anak. Sekurang-kurangnya 3 orang anak dia akan major. Sebab dia patient. So, jadinya, macam mana doktor? Saya cakap, macam tu lah. Saya kata, kalau saya cakap jangan kahwin kan, lagi kan, masalah saya nanti. So, saya kata, saya bagilah the scenario and the risiko dia. Dia dah bersama saya lebih daripada 10 tahun. Dengan club kan, dengan NGO. So jadinya dia mesej saya, dia tanya, macam mana ni doktor, saya nak kahwin ni tapi saya nak anak. So kurang-kurangnya 2 pun cukup lah doktor. Oh sudah. Saya kata macam mana ni, saya kata. So jadinya, lepas tu dia mesej saya, doktor saya tak jadi kahwin lah, saya kata. Okay. So, dia putuskan perhubungannya, sedih tu. Kak dah siap. Paksalah batalkan perkahwinan. Dia mesej saya lagi, selepas 2 tahun tu. Dia mesej saya, doktor saya nak kahwin ni. Lagi berapa bulan? Saya kata, 6 bulan. Hai, kenapa 6 bulan? Kenapa tak setahun? Saya kata. Lepas tu saya pun takut nak baca diagnosis dia. Alpha. So alpha dengan betanya tak apalah. So boleh kahwin. Lepas tu dia kata, saya baik doktor kata boleh kahwin. Kalau tak boleh kahwin, saya tak nak kahwin sampai bila-bila. Oh sudah. So kita pun takut menjadi sebab yang kita budak tu tak berkahwin. So they happy after that. Okay. So the decision is made by you. Okay. Ada juga patient kita, patient sama patient berkahwin. Dah saya cakap berulang kali. Dah lebih 10 tahun aku kenal dia. Lepas tu saya kata, kenapa kahwin? Saya kata, saya cinta sangat dekat dia. Tak apa doktor, kita sanggup bersusah payah sama-sama pergi hospital. Awak ingat darah awak sama ke dengan dia? Saya kata, it's okay doktor, saya hantar dia. Lepas 1 tahun dia berkahwin, bergaduh terus. Anaknya dilahirkan, anaknya pun major. Of course lah. Lepas tu duit lagi. Dengan keluarganya tidak berapa senang. Masalah duit lagi. Akhirnya bergaduh terus. So bila dah hilang daripada radar. So saya call tak angkat. Sudah. Is that happen? Sebelumnya bercakapnya, okay tak apa. Selepasnya adanya anaknya sentiasa sakit, dia pun sentiasa sakit. So what happened? That's a problem. Okay, the things is must do the awareness first. So then do the public population screening. Saya rasa di Indonesia masih lagi tak buat tu. It's very lacking and banyak study yang boleh dilakukan. Okay, then genetic counselling. Itulah saya kata, bila kita buat genetic counselling pun, tak apa, you tak payah berkahwin, you ambil anak angkat. Tak nak, saya nak anak saya sendiri. So jadinya itulah. Kita kadang-kadang bila dah married, susah kita nak kata jangan ada anak. Ada pasien saya, nombor 1 anak thalassemia, nombor 2 anak thalassemia, nombor 3 anak thalassemia. Saya kata kenapa sampai jumpa anak nombor 3 sebab saya nak anak nombor 1. Lantasnya anaknya nombor 4 tu normal lah tapi pembawa. Tapi okey lah, sihat. Itulah the things yang kita hadapi setiap kalinya. Okey, di Malaysia, okey, ini yang saya cakap tu, dia dah start daripada umur awal dan kita boleh elak daripada perkahwinan. Okey, daripada perkahwinan pembawa dengan pembawa. Okay, it's very important awareness. You dah tahu dah saya cakap hari ni. You bercerita dengan lebih orang kawan you, 5 orang. You bercerita dengan keluarga you. Keluarga you share dengan orang lain. That's the awareness yang kita boleh buat di kalangan keluarga untuk elakkan perkahwinan. Okay, di Indonesia, okay, Indonesia we notice that they have the 3 phase, okay. The phase is phase 1. We are doing the family screening program. If that the patient are already detect have the thalassemia. Contohnya, anak dia ada thalassemia. So, kita buat screening dekat semua ahli keluarga dia dan semua rakan keluarganya. So, untuk elakkan perkahwinan selepas itu. Tetapi, kita tak buat secara broad. Kita buat bila adanya thalassemia patient kepada keluarga itu. So, kita nak tahu ada berapa carrier dalam tu. So, then after that, kita the phase tu dia extended to the family, okay, the public, tetapi tidaklah require before marriage. Okay, when it's not required before marriage, okay, biasanya orang kita ni tak mau buat lah, kan? Malas lah. Apa tu thalassemia tu? Apa itu? Tak ada apa. So dia hanya tahu HIV saja. Kahwinannya sebelum harusnya HIV. Kalau tak buat HIV test di Malaysia tak boleh kahwin. So jadinya tak buat thalassemia. So after yang dia dapat anak thalassemia, okay, that have the start of the problem. Bila melibatkan duit, itu adalah paling besar isu. Okay. So culture juga. Malu. Okay. Pernah ya anak patient saya makan kelawar hidup, dibelahnya kelawar. Dia ambilnya hati kelawar tu, dia makan terus. Saya kata, kenapa makan? Boleh naikkan HB, doktor. HB ke kematian ni, saya kata. Dia kata, okey doktor, ini kita punya, saya dapat, dia kata bagi tahu kalau makan hati kelawar boleh terus sembuh. Bukan terus sembuh ni, terus ke liang itu. So saya kata, we need to save the children. Okay. Kita kena lahirkan, kalau boleh kita cuba elakkan iaitu lakukan saringan. It's very important. You just, all of you remember, do the awareness before, do the screening before marriage. Janganlah dah lagi 2 bulan nak kahwinnya, buat screening. Lakukan di umur yang awal. Umur 21, 22 tahun yang masih belum bercinta lagi. Dia kata, tak apalah, tak apa. Waktu itu manis semuanya. Kalau selepas itu balik botol lah habis. Bergaduh, berentam terus, ya. Sebabkan duit. It is very masa kena hantar anak cuti. Seminggu kadang-kadang demam saja. Because of the reaction. So jadinya siapa yang akan jaga anak? So diorang akan start bergaduh. So menyebabkan perceraian. Itu yang kita nak elak sebelumnya berlaku. Perkara-perkara seperti ini. Bila dah berlakunya perceraian, what happened to the kids? Yang ada sampai 3 orang anak. Itu is very, very pity to them. Siapa yang nak jaga mereka? Itu sangat sedih lah. So, you are the one can save the children. Okay? Stop thalassemia and do the screening. And it's kalau di Malaysia ni RM200. Kalau anak-anak sekolahnya percuma. Kalau yang dewasanya yang berkahwin, RM200 di private hospital. Tetapi it's okay lah, RM200. Bermaknanya akan keluarkan duitnya setiap bulannya satu pesakit RM1,000. Kalau satu pesakit thalassemia di cover oleh kerajaan Malaysia adalah RM1 juta. RM1 juta. Kerana ubat pilnya, SJ-nya berharga RM700. Harus makan 10 biji. Itu saya kata sekali you makan lobster ni. Saya kata lebih mahal daripada lobster ni. Lepas tu dia buang pula. Dia tak nak makan. Itulah. Okay, stop dari sini. Ya. Okay. Okay. So, what you want to feeding your children? Pilihlah. You nak beli susu atau nak beli darah. Darah je harganya 500 satu beg. Susunya harganya 1,000 gram baru sekilogram baru 100,000 ya. Harganya beza ya. 100,000 yang 500,000 harus makan sekali. Tahannya 2 minggu. Gimana tu? So jadinya itu yang kita perlu elakkan. Okay. So then ada. So share. Berkongsilah di mana-mana media. Okey, sedikit ilmu yang kita kongsi memberi manfaat kepada orang lain. Okey, barakahnya lebih banyak dan lebih besar. Kalau di So sebarkan. Okay, after that. Kenal ini. Pak Mael ya. Abang Mael ya. Upin Ipin itu. Upin Ipin tu kata 1 dari nisbah 20 rakyat perlu adalah pembawa. Tetapi di Indonesia 1 nisbah 10 adalah pembawa. Disebabkan tak adanya awareness di sini, okey, menyebabkan meningkatnya 1 nisbah 10. Okey, peningkatan tu berlaku setiap tahun dan kita nampak ia tidak berjaya. Okey, lakukan saringan. Okay, test thalassemia before choosing. Before choosing a life partner. Bukan after choosing the life partner. After choosing the life partner, bergaduh. Terus. Jangan. Takkannya. Tak apa, kita boleh tukar-tukar nanti. Tak apa, kita boleh bawa pergi hospital sama-sama. Tak apa, it's okay. Kita kan kerja, kita ada duit. No. It's very increasing the every expenses every year. Meningkat setiap tahun. Kalau dulunya passion kita diberinya ubat mahal. Sekarang kita terpaksa kurangkan ubat yang murah dan pakai mesin. Tetapi it's about the costing. Pesakitnya makin ramai, kosnya makin rendah. So jadinya kita terpaksa bagi ubat yang tak berkualiti, akhirnya menyebabkan complication. That's the thing. Okay, saya rasa itu saja daripada saya. Thank you. Saya boleh buka sikit video ke kalau boleh?
Alright, really cool. So we have a video.
Apa nak?
Patient yang saya kenal sejak kecil.
Sakit ke?
Itu mesin desferal tu di badan dia. Dia harus pakai mesin desferal
Sakit
tu
perut?
setiap hari
Apa?
selama 6 hari. Dan rehat
Baik kan?
selama 8 jam.
Bi,
Dan anak yang
masih tidur
3 last.
sakit perut ke? Kat mana sakit ibu? Sini. Tapi tak bengkak, ubat jalan.
Itulah beg yang dia ada mesin desferal yang dia letak dalam beg tu.
Ibu,
Kalau ke sekolah pun bawa beg tu. Harus diletak di bahu, bawa beg. Tengok dia cucuk jarum tu memang tak boleh tanggal selama 8 jam. So 8 jam dia akan pakai mesin tu. Kadang-kadang dia cucuk
Di jarum yang elok tak sakit
Andainya hatiku bersuara, pastinya ia akan berkata, cukuplah sayang, aku tidak berdaya.
Ini anak dia tidur.
Andai kau merasakan sakit yang kau berikan kepadaku,
Hari-hari itu ada konsultasi dekat anak dia nombor 2.
yakin kau tak
butterfly tu kita beli harganya RM4. Dia pakai ni.
Berkorban nyawa, tanda kasihnya, berkorban raga. Sebenarnya perjalanan itu
Terus aku tunjuk tu
melukai
jadinya sebabkan inilah dulu kita macam budak-budak
sememang
dia akan terima desperate, tetapi after 10 years dia pakai desperate badan akan reject, so dia terpaksa guna ubat lain. So dia menyebabkan dia tak nak reluctant nak pakai ubat. Bila dia reluctant pakai ubat, akhirnya dia akan menjuruskan diri dia ke kematian. That's why lah. Terima kasih.
All right. Thank you so much, Dr. Isma. It's really cool that I think that it's really hit my face, right on my face, because the awareness in Indonesia is really low. And then we learn a lot from the Malaysian perspective, from your research as well. So yeah, the importance of this, the awareness, the transfusion of thalassemia, and then we learn a lot that even in Malaysia, they have the association, the organization, the NGO, the club, and everything. And I think that we don't have that much. I don't know, and I have no idea about this topic as well. But yeah, it's like on my face to see the cases and everything. So yeah, so everyone, this is a good chance for you. Really good opportunity for us to learn from Dr. Isma. Don't forget to give your questions on the chat box here. So we'll learn from the Malaysian perspective on the good practices and the research as well. And yeah, we have some similarities, but we have differences as well in the prevention and everything. So because I could say that in Indonesia, the awareness is less compared to Malaysia. And I think that another key point is that the policy making, like the enforcing that it doesn't happen in Indonesia that much. So, and the message that Dr. Isma already shared to us about sharing about the awareness. You can share to all of your platform, the social media and everything, because one message, yeah, it can be give us a really good positive impacts, and the multiplier effects will be really cool and save the children for our better generation. Thank you so much, Dr. Isma. It's a really cool session. And yeah, this is the end of the first session with Dr. Isma. We will have the Q&A in the end of the session with our three speakers, Dr. Isma. So we will keep the questions until we have the last speaker. All right. So moving on to our second speaker, we have our beloved lecturer here, the lecturer of Blood Bank Technology Program of Stikes Guna Bangsa. Yeah, we have Mrs. Gravinda Wideswara. So let's have some CV of Mrs. Gravinda. So because our program of Blood Bank Technology would like to present one of our lecturers' research, and we decided to share one of them, Mrs. Gravinda. So she is our Blood Bank Technology Program lecturer of Stikes Guna Bangsa, Yogyakarta. Mrs. Gravinda completed her Bachelor of Sciences in Biology, as well as with her Master in Biology as well at Universitas Gadjah Mada, Yogyakarta. And her research field are on the transfusion-transmitted infection, microbiology, and biology cell. So before teaching at our program, at our Blood Bank Technology, Mrs. Gravinda also gave classes and a lecturer at Medical Laboratory Technology from 2019 and 2022. Okay, without further ado, let's have our second session with Mrs. Gravinda. Over to you.
Oke, thank you, Mr. Wisnu. Apakah suara saya terdengar? Okay. First of all, Assalamualaikum warahmatullahi wabarakatuh. I want to say thank you to Stikes Guna Bangsa Yogyakarta, especially Blood Bank Technology Program Studi that give the chance or the opportunity to me to share my research about the analysis of bacteria. But before I start to share my research, I want to say sorry because I'm not in a good condition, I think. Serak-serak basah gitu ya, seksi gitu. But I want to try my best to share my research about, okay, let me share the screen. About the analysis of bacterial growth in the transfusion transmit infection laboratory at PM Sleman Regency, the best of the one best of the Red Cross in Yogyakarta, Indonesia. So this research is collaborate with PMI Sleman Regency with Dr. Suhartono. And I want to say thank you to PMI Sleman Regency also because PMI Sleman give me the time and the room, the TTI laboratory to make this research happen. Okay. So I start with the introduction. Okay. The blood is a therapeutic products and must be taken in accordance with the blood supply unit's quality management system to ensure their quality and safety, and also to minimize the potential for contamination by bacteria or other microorganisms. Like Dr. Isma said before, that blood is a medicine, I think, yeah, for the thalassemia patients. Also in this session, blood is a therapeutic product. So we have to make this product, blood product, minimize to contamination by bacteria. So what is bacterial contamination? Bacterial contamination of blood products can come from within the donor's body or from the environment, especially the air. Then environmental contamination can be obtained from the process of or collecting donor blood, the process of making components and storing blood, and also when the distribution of blood. Environment contamination, the one is transmitted transfusion infection laboratory. And the Ministry of Health of Indonesian Republic in 2015 said that standard of blood transfusion service had a step that the room and equipment used for processing blood components must have the quality management system with the aim of eliminating risk. And this risk include bacterial contamination, exchange of blood products, like what I said before, transmission or of disease, or unexpected side effect due to the use of blood components or when transfusion reaction. Jadi kontaminasi bakteri itu bisa terjadi ketika proses pembuatan komponen darah, ketika ketika pasien mendonorkan darahnya atau ketika distribusi darah kepada pasien itu sendiri. Sehingga in this laboratory of transmitted transfusion infection, where is the room used for screening tests such as HIV, hepatitis B, hepatitis C, also syphilis, have to free from all kinds of mechanisms. Yeah, not just bacteria, but also viruses and plasmodium and etc. Okay. Very important. This research that I used to make the research good, I think, yeah. Oh yeah, the slide. Moving. The next slide. Okay. From Rahman et al. In 2011, the research in Ghana Temple Female Teaching Hospital, out of 18 bags of donor blood stored in the refrigerator, 14 bags or 17.5% were contaminated with bacteria. And also in Obevemi State Teaching Hospital, Nigeria, from from 160 cc, whereas 14 blood bags or 8.8% were contaminated by bacteria. And then from Aksi et al. 2019, where is bacterial contamination, 18 blood bags, 14 bags of blood contaminated with gram-positive, and 4 blood bags contaminated with gram-negative. And then Wilkrania et al. 2019 also said about the bacteria are often found in the environment outside and contamination from inside room, because bacteria have the ability to adapt and adjust to various mental conditions like air, material organics, and then insect, animal, human vectors. And there are Ramatullah et al. In 2021 and Ramatullah et al. 2022 said about the 10 petri dishes used as example contain contamination in medium Na with a result of 20% contain gram-positive, 70% contain gram-positive Coccus bacteria, and 10% contain gram-positive Bacillus and Coccus bacteria. Also in media McConkey agar with result 16% contain gram-negative Bacillus, and 30% contain gram-negative Coccus bacteria, and 10% contain gram-negative Bacillus and Coccus bacteria. And the last result, Ramatullah 2023, there were 20 samples taken from 5 points in the transmitted transfusion infection laboratory at PMI Yogyakarta, was found out that bacteria growth in media Na or MCA with details. 4 samples gram-positive Bacillus bacteria, 11 samples from gram-negative Bacillus bacteria, 5 samples of gram-negative Coccus, and 1 sampel of gram-positive Coccus bacteria. Jadi dari semua penelitian yang sudah dilakukan itu menemukan bahwa banyaknya kantong darah yang terkontaminasi oleh bakteria. Tidak hanya kantong darah dari whole blood, tapi juga packed red cell maupun dari TC. So, in this research, purpose is to analyze the growth of contaminant bacteria that grew on Na media and MCA media or McConkey agar cultures in the transmitted transfusion infections laboratory at PMI Sleman Regency. Jadi kami di sini ingin menganalisa bakteri udara apa khususnya yang mengkontaminasi laboratorium TTI atau in bahasa infeksi menular lewat transfusi darah. This is the method. This type of research is true experiment. True experiment is a research method that is truly experimental because the researchers, we can control all the variables that influence the course of the experiment. So we control the temperature, the pH, pH, and etc. Okay, this is the procedure. So we carry out by placing petri dishes containing Na and McConkey media agar in the transmitted transfusion infectious laboratory at PMI Sleman Regency, and then we incubation and bacterial identification processes were carried out at the microbiology laboratory of Stikes Gunabangsa Yogyakarta. Samples for this study were Na and MCA culture media, which were placed at 5 points in the room. In this picture, there are one place the Na and MCA media under the blood group confirmation tool, and then second place Na and MCA media placement position near semi-luminous immunoassay or CLIA equipment, and then third place position of Na and MCA media next to the blood bank for quarantine blood, and then the fourth place the Na and MCA media on the table near the centrifuge, and the last place MCA media position under the SimDondar application computer table. Besides that, the temperature on the 37 degrees Celsius for Oh sorry, ini diinkubasi ya, diinkubasi pada temperatur 37 setelah melakukan pengambilan sampel for 24 until 48 hours. Okay. And this is the procedure. We prepare the required media in Petri dish, and then media is opened and placed in the laboratory of PMI Sleman for 30 minutes. Then media is taken and we bring it to microbiology laboratory of Stikes Gunabangsa Yogyakarta. Then the media is incubated at temperature 37 degrees Celsius for 24 until 48 hours. And then we do the gram staining was carried on Na or nutrient agar media and McConkey agar media. So this is the result of our research. Double one is about bacteria growth on Na media. Yeah, we can see that sample point 1, 2, 3, 4 until 5, there are colonies bacteria that grow. The total colony bacteria in sample 1 is 93 colonies with gram staining red color and morphology rod shape and lined up. And the result is negative gram bacteria. Also in sample point 2, 3, 4, 5, there are gram staining red colors and the result is negative gram. Morphology bacteria rod shape shingle for sample 2, and then for sample 3, the morphology is rod shape and lined up. And then for sample 4, there are rod shape morphology bacteria and single. And sample 5, there are rod shape bacteria morphology and single. And this is the table 2 about bacteria growth on McConkey agar media. From sample 1 until 5 in MCA media, also there are red color gram staining. So the result is negative gram bacteria. And the shape of sample 1, there are rod shape and lined up bacteria. And sample 2, there are rod shape morphology bacteria and single. Sample 3, rod shape bacteria and lined up. And sample 4, rod shape single morphology. And sample 5, rod shape and lined up morphology bacteria. Based on table 1, the results showed that on table 1 and table 2, the results showed that on nutrient agar media or McConkey agar media, there was bacterial colony growth at 5 locations in the transmitted transfusion infection laboratory of PMI Sleman Regency. And from the result of gram staining, both on Na or MCA media, the bacteria were found to be red with a rod-shaped bacterial morphology, most of all, yeah. And then the red color proves that the bacteria found are gram-negative bacteria. From the result obtained, the bacteria that grow are red, have a rod morphology, short, spray rod. So it's so suspect that the bacteria that grow belong to the Enterobacter sp group. Nah, there are several possibilities for finding Enterobacter sp or Enterobacter species in the transfusion transmitted infection laboratory of PMI Sleman Regency room. This could be because at the time of sampling, the CPOB system hadn't been implemented at PMI Sleman Regency. But now, the PMI Sleman Regency sudah memakai CPOB atau CPOB. Oke, saya agak lupa ya CPOB kepanjangan dari cara pembuatan obat yang baik. Oke ya. And then the result cannot be confirmed because the analysis of bacterial growth only reaches the gram staining state and hasn't yet reached further identification. However, the result of the research can be referenced to be used as a consideration for prevention or treatment in order to minimize bacterial growth in TTI laboratory of PMI Sleman Regency. Okay. In general, the characteristic of Enterobacter sp. This include gram-negative bacteria, rod shape, live motifly, can live at an optimum temperature, can live in aerobic, anaerobic condition, and in general, this group of bacteria lives in water, soil, waste, and can live in the human digestive tract. So, Enterobacteriaceae are normal flora in the human intestinal tract, and this bacteria also easily carry genetic material through horizontal gene transfer. Okay. Now, several types of bacteria belonging to Enterobacteriaceae group like Shigella sp, Escherichia coli, Salmonella sp, Enterobacter proteus, Klebsiella sp, and Aeromonas sp. So this is our conclusion. Based on the research conducted both on NA media or MCA media, it was found that the growth of air bacteria in the TTI laboratory of PMI Sleman Regency with bacterial characteristic leading to the Enterobacter sp group. Meskipun setelah ini juga masih harus dipastikan dengan identifikasi bakteri. Okay, this is our reference that we use in this presentation. Okay, thank you. And I give it back to Mr. Wisnu.
All right. Thank you so much, Mrs. Grafinda, for sharing your research. It's really cool that we learn a lot about the TTI, right? The transfusion transmitted infections. And it's really practical sharing and practical experiences because Mrs. Grafinda conducted the research at one of the best Red Cross Indonesia in our province, which is in Sleman Regency. And this research also has been published.
Not
Not
yet,
yet.
in October
In October.
2025.
So we will share once it got published, and then we will share the research. So, dear participants, it's really cool and to have the sharing session and research sharing from Mrs. Grafinda, specifically on the TTI. So if you guys have any questions, feel free to drop your questions through the link that we have provided on the chat box. So we will get back to our speakers later on. All right. Moving on to our last speaker today. The third speaker is really cool that we have Dr. Fitri Nadifa. And because, yeah, she is one of the best lecturer at our campus. And Dr. Fitri is a lecturer at Medical Laboratory Technology program at Stikes Gunabangsa, Yogyakarta. And like Mrs. Grafinda, that she is also expertise in biology. Like Dr. Fitri earned her bachelor, master, and PhD in biology at Universitas Gadjah Mada, Yogyakarta, and the field of the study, of course, in biology and microbiology as well, and also the medical biology. Okay, wait a moment. And yeah, in molecular biology, microbiome, and clinical microbiology, and her latest research is talking about the characterization of the urogenital microbiome in patients with urinary tract infections. So, without further ado, let's have our last session with our last speaker, with Dr. Fitri Nadifa. Over to you.
Yeah, thank you, Mr. Wisnu. Can you hear my voice clearly? Okay, thank you. Let me share first my presentation. Okay, wait a moment. Okay, can you see the slides?
Yes.
Slideshow. Okay. Thank you. Good afternoon all. Assalamualaikum warahmatullahi wabarakatuh. Yeah, thank you for the time. In this very good opportunity, I want to share a little bit of what we call the most basically molecular method that already been used globally. It's about polymerase chain reaction for bacterial identification. I am Fitri Nadifah from Stikas Guna Bangsa, Yogyakarta. I'm the medical laboratory technologist from the years of 2009 in Stikas Guna Bangsa, Yogyakarta. So, previously, Mrs. Gravinda already mentioned the topic about transfusion transmitted bacterial infection. She's the expert in this topic, so I don't want to talk much about this because she's already mentioned lots about the contamination of some microorganisms like some bacteria or some parasites or other like virus in blood products. So she already mentioned that typically this TTBI typically involves the introduction of bacteria into the bloodstream through the contaminated blood products during transfusion. So the bacterial contamination can occur at various stages during collection, processing, storage, and or transfusion. So the common source of bacterial contamination is like from skin flora, collection or storage equipment, contamination, and improper handling procedures. We can say that the spectrum of pathogenic organism varies based on the collected blood product. And the most prevalent bacterial pathogens isolated from the platelet products is like Streptococcus viridans, Streptococcus bovis, and beta hemolytic streptococci with an incidence of 39% to 48%, while coagulation negative Staphylococcus with an incidence of 21 to 24%, and also Staphylococcus aureus, it's also have incidence of 4% to 9%. While the RBCs, one of the blood products, it's mostly contaminated with Yersinia enterolytica bacteria, Pseudomonas, Enterobacter, Serratia, and Escherichia coli, as well as the Babesia microti, that it is the most frequently associated organism with fatalities after RBC transfusion. So we keep moving to the key points of this topic about bacterial contamination in the blood products, and what laboratory can do to diagnose this is to do some standard or gold methods, that is culture-based testing. But this diagnosis result from this culture-based testing is resulting in days to weeks. So this is the weakness of this method. Sometimes that bacterial or other microorganisms that we grow in media culture, sometimes it cannot grow. It makes some other weakness of this method. So we are looking for the other method that maybe it can be the complement of this culture result that we call it as polymerase chain reaction, one of the molecular method that already widely used globally. But first of all, we have to know in bacteria, we have to know what genetic materials resist in the body of bacteria. We can see from the illustration right here in the left, the DNA of most bacteria is contained in the form of bacterial chromosome that contain its a singular circular molecule ya. Jadi ini adalah sebuah gambaran tentang bagaimana material genetik yang ada di dalam bakteri. Yang pertama adalah dalam bentuk bakterial kromosom. Jadi dia adalah sebuah molekul DNA yang berbentuk single circular. Kemudian ada juga yang disebut nucleate. It's an irregular shape structure contained chromosome, also protein as well as RNA. And then the mostly known from bacterial, its material genetic is plasmid. It is a small circular DNA molecules that contain mostly genetic gene that yang membawa sifat bakteri ini dalam memproduksi protein. Before that, we have to know, this is so basically in biology, student of biology or other medical fields should know about the structure of DNA, deoxyribonucleic acid or DNA. So this is the molecule that carries genetic information for the development and functioning of an organism. Not only in human or animals, plants, and other bacteria also contain this DNA since they need protein to so they can live ya, bertahan hidup. The DNA is made of two linked strands that wind around each other to resemble a twisted ladder. Jadi seperti anak tangga yang berputar. It twisted. A shape known as a double helix. This is the illustration of the double helix of DNA structure, which is found in 1668, yeah, long time ago. This is also illustrated how exactly the structure of DNA. So the H strand has a backbone made of alternating sugar or deoxyribose and phosphate groups. And then it attached to each other is one of four bases, consists of adenine, A, cytosine, C, guanine, G, or thymine. The two strands are connected by chemical bonds between the bases. Adenine bonds with thymine, so A with T, and cytosine bonds with guanine, C with G. And then the sequence of the bases along DNA's backbone encodes, this is the most important, it encodes biological information such as the instructions for making a protein or RNA molecule. And then we talk about the bacterial gene. So what is a bacterial gene that mostly resists in plasmid circular DNA that we talked about it previously? Bacterial genes are functional units of DNA that have their effects through diffusible products or non-essential replicants, which often determines resistance to antimicrobial agent, production of virulence factors, or other functions. So this is the picture or illustration how a gene is actually a genetic information to make a protein. But the question is, if we had a sample suspected containing some bacteria, then how we get the DNA? So we can get the DNA with the procedure of DNA isolation. It is the separation of DNA from other molecules in cells. And humans or bacteria, viruses also can be isolated. So in bacteria, chromosomal DNA and plasmid DNA, that is, we can harvest cell DNA. This is the picture illustration. Bacteria, we can get the DNA through the DNA isolation, and then we can purify them with the procedure of DNA purification. Many protocols mention about the DNA isolation procedures, but basically we have to do some steps that including the first one is we can culture or cell harvesting if it requires, but in case if it can be cultured through harvesting, a higher number of cells are obtained for isolation. And then we have to do the bacterial lysis. Lysis will break the cell wall and releases the DNA using some reagents like maybe detergent contain reagent or alcohol, some alcohol. And then we do the DNA isolation. The DNA isolate is collected in a separate tube and precipitated using alcohol. And then we do the DNA purification. This is the most abundant way for DNA purification. But however, we can use some kits that also be to purify bacterial DNA for sensitive experiment like DNA sequencing. And then the last steps, DNA can be eluted in an elution buffer using the buffer or deionized water. This is the example of procedures about the harvesting cell from the bacterial culture through the conventional culture. The first step is we grow the bacteria, or we the example in this is Bacillus subtilis. We grow them in nutrient broth and then incubate them overnight, and then we harvesting them not using centrifuge. The centrifuge method will break the wall of the bacteria, so they will lysis the bacteria cell wall. I'm sorry. And then we waste the supernatant, and we can harvest the pellet that is that we use in the next step. This is an example of purification DNA using a mostly wide known of kits to purification from Kia kits. So they have their own procedures to isolate the DNA from some example like stool or from bacterial culture. Many researchers use this procedure from Kiam region. So later, to identify bacteria, we must know which genes play a role in bacterial identity. The 16S rRNA gene, it has been the most widely used gene for this application because it presents in all bacteria and has conserved regions. But identification can also be done by identifying genes that determine the pathogenicity of the bacteria. For example, in INVA, SPIC, and TIPD in Salmonella, we can also this gene for the identification. Once we have found the target gene, we need to amplify the gene so that it can be easily visualized later. So what is the method we use here? We amplify the target gene using polymerase chain reaction. or PCR. The question is, what is polymerase chain reaction? So this is the technique for producing millions of copies of short DNA segments through the repeated cycles consists of pre-denaturation, and then the main step, which is denaturation, annealing, and elongation or extension. This is the basic principle of the amplification of DNA segment or gene target. And like we cooking something, we do the PCR by mix the reagents. We need to do this method. So in a stereo microfuge tube, the following reagents are mixed. First of all, of course, the genomic DNA template with about 50 nanogram until 250 nanogram. Kemudian we need deionized water, primers. This is also important and cannot be inert. It's very, very important. Reverse and forward primer, and then the DNA nucleotide base, DNTPs. Tag DNA polymerase, it's also very important, this enzyme polymerase. And then the tag aside buffer contains 3 HCl, MgCl2, and KCl. This is how the PCR machine works here. So they do the first step, the denaturations. That the reagents are heated to 94 degrees Celsius for 3 until 5 minutes. So the high temperature causes the hydrogen bonds between the bases to break and the strand separate, memisah ya, jadi dari yang double helix menjadi single helix, separate into 2 single DNA strands. And then the annealing steps. In this step, the reaction is now put to didinginkan. The temperature is lowering down. It's about range-nya 50 until 65 degrees Celsius for 10 to 30 seconds. Quick time. This step allows the forward and reverse primers to attach, menempel to the single-stranded DNA yang sudah kita pisahkan tadi through hydrogen bonds. And then the third step is elongation or extension. In this step, the reaction now is heated again at about in 72 degrees Celsius for 5 to 10 minutes. In this step, this is an optimal temperature for tachia. This is from the isolated from the bacterial Thermus aquaticus. Researchers isolate the polymerase DNA from this bacteria. To attach to the primer and start adding the nucleotides one by one in 5 to 3 direction to build the complementary DNA strands. This is the benefit of the PCR method. So from the single strand DNA, we can amplify from 2 to 4 to 8 until millions of copy of DNA. So the name of this method is polymerase chain reaction. Like this, from 2, we can yield 2 to 4 to 8, and so we can get the millions of copy of DNA. Maybe we can see clearly about the basic principle of PCR. Through this animation, we can see, I want to play the video. Dengar. Yeah, this is the basic information about the PCR. Oh, I'm sorry, there is technical problem. I want to stop it first. Wait a moment. This is the video of PCR explanation from Sumana's ID. Oh, maybe some other technical problem. I'm sorry. This is explanation of the how it polymerase enzyme works in this step. This is ini from the double helix. Because of the heat, it becomes single strands, and then the primers attach in one side of the single strands. Because there are hydrogen bonds between them. We can see that adenine always make a bond with thymine. When we rise the temperature, the polymerase enzyme started to work, and then build the material in this single strand to make double strand. The yellow one is polymerase DNA. After that, we starting again the cycle from the denaturation, annealing, and elongation. Jadi ini mulai lagi ke proses denaturation, and then starting again until 100 of cycle, we can yield many copies of DNA. So this illustrated how the copy numbers of target DNA increasing. It's just seperti kita main-main dengan suhu, kita dapat menghasilkan berjuta-juta kopi dari DNA target kita. Like in this illustration. Millions of copies. And in this microcentrifuge tube. Okay, thank you. For the next. I want to share again the next slide. So this is the illustration how researchers works with a PCR. Before using the PCR machine, researchers has to design and order primers, and then mix them and load them into the PCR machine. Like this, researchers making the PCR mixture, and then loading them into the machine, and the PCR will works it cycles that consist of denaturation, kemudian annealing, and then the elongation steps. How we can visualize of PCR results is by electrophoresis. So once PCR is completed, gel electrophoresis is used to check for quantity and size of the fragments. Gel electrophoresis is a method where the DNA fragments are drugged through a gel matrix using electric current to separate the DNA pieces according to sizes. A standard DNA ladder is included so the DNA sizes can be determined. So this is how we can identify the DNA from the sample we analyzed previously. And then this is the illustration of an agarose gel electrophoresis setup. The agarose gel sits in a tank of buffer. The sample mixed with loading dye are placed in wells at one end of the gel. And then an electrical current is applied, causing the negatively charged DNA to move towards the positive electrode. This is the example of the PCR result that we visualize using gel electrophoresis. Because of the dye, jadi pewarna yang bisa memandarkan fluorescent, we can see the bands ya, pita-pita DNA yang sebenarnya itu adalah gen target kita jika memang muncul atau appears. Of course we have to harus safety ya. Jadi untuk melihat hasilnya ini harus dengan alat yang memang digunakan untuk visualize the PCR results, or we can use the glass to safety visualize or see the results. When the samples have run far enough to obtain sufficient separation, the gel is removed from the tank and placed on a UV light box. The intercalating dye then allows the sample bands to be visualized and their size determined in comparison to a DNA ladder with known band size. So the far left in here is the DNA marker or DNA ladder. It's for the standards or to show us kira-kira berapa panjang atau berat dari pita DNA yang muncul in this gel electrophoresis. This is another sample of the PCR results from the electrophoresis visualization. Single gene di sini muncul ya dalam bentuk pita DNA bands. We can actually mengira-ngira ya, based on the marker, the DNA bands is maybe it's about 1,400 base pair, the length of this gene. So the size marker that is used as a reference for the length of the sample DNA fragments is in this far left of this gel. When a DNA band from the target gene appears, it means that the sample we are analyzing is positive because the pita DNA or DNA band is muncul. Jadi bisa kita katakan bahwa dalam sampel yang kita analisis itu ada gen target kita, target gene. And then these results can also be further analyzed using DNA sequencing technology and match with a data bank internationally. For one example is an CBI, or we often call it as gene bank. So that's all about the basic or conventional PCR, but I want to show here some PCR application PCR, both conventional and applied, has been widely applied in various health sectors. This is some examples from the articles from Al-Khatib, applying RT-PCR standard detection procedures of HPV and HCV in Wasit blood bank. And then here from Dreyer et al., the real-time polymerase chain reaction in transfusion medicine, the applications for detection of bacterial contamination in blood products. And then this is from Wellinghausen et al., diagnosis of bacteremia in whole blood samples by use of a commercial universal 16S rRNA gene-based PCR and sequence analysis. And the last one is from Leo et al., which is detecting the presence of bacterial DNA by PCR, can be useful in diagnosing culture negative cases of infection, especially in patients with suspected infection and antibiotic therapy. So there is a wide opportunity to use this molecular method, the conventional PCR or other the applied, to do some research works, especially in blood transfusion. So that's all. Thank you. I want to give back to Mr. Wisnu. Thank you.
All right. Thank you so much, Dr. Fitri, for sharing your research. It's really cool that, guys, we learn a lot specifically for you from the medical lab students that we got a lot of knowledge and information about the molecular method, which is PCR, and specifically on the bacterial contamination of blood products. So it's a really good combination that Dr. Fitri talked about the blood products in the midst of the PCR on the molecular method. So yeah, it's a good opportunity for like a blood bank technology students and also medical lab to learn a lot from Dr. Fitri. Thank you so much, Dr. Fitri. All right, this is the moment that you're waiting for to have a discussion with our speakers, our guest lecturer as well. So we would like to show the questions. We received so many questions, Dr. Isma, and each of the speaker will have 3 or 4 questions, and we have already summarized the questions here. Okay, let's go to the first question. Wait a moment. So yeah, the speakers on our Google form exploded but because we have a limited time, so we will like only have 3 or 4 questions for each speakers. Okay. All right. So the first question is coming from Mahmuda from ABKPMI. So the questions for Dr. Isma. Apakah penyintas talasemi bisa mempunyai banyak low antibody karena sering melakukan transfusi darah? Can thalassemia survivors have a lot of low antibodies because they often undergo blood transfusions?
Pada Mahmudah.
Yeah.
Okay, di atas pertanyaan soalannya. Yes, of course, the patient that always transfuse the blood, okay, for sure they have will develop of the allo antibody in their blood. That's why we have the graded of the seriousness of allo antibody in their body, because the grade 1, grade 2, grade 3, grade 4, which is afterwards, if they are already reached to grade 4, they have to grade 3 or grade 4, they need to sign off the consent form. That's the responsibles to the patients if anything happen towards the between the transfusions. So development antibody is every month when they are receive the blood back. That's why that I told before that I need to do the antibody and identifications to prevent the allo antibody forming for in fast reactions. Okay, thank you.
All right. Thank you, Dr. Isma. So the next question. Okay, this is from Mariana Placidia Bali from Stikes Gunabasa, Yogyakarta. So as a first semester physiotherapy student, I am interested in finding out how biomechanical principles can be applied in designing exercise programs for thalassemia major patients who experience limited movement in the hip joint. Is there any research comparing the effectiveness of manual therapy with therapeutic exercise in reducing joint pain in thalassemia major patients who experience osteonecrosis?
Okay, terima kasih atas pertanyaan daripada Mariana. Okay, salah satu the complication of the patient is osteoporosis because they are lacking of the calciums inside the body. Because we notice that the productions of the blood bag is caused from the calciums. It's one of the agents, okay, called like agents lah to produce. That's the one of more things because the patients who are you cannot be stands in a long times because they are very kata not in very good conditions pinggangnya kalau during the transfusions, okay, they have to the bed ridden, okay, bed rest, okay, because it's long time to wait, okay, they have a problem with the back tulang belakangnya akan ada masalah. So jadinya it's very good, okay, when you do the opportunities to expose what the kind of exercise, okay, that can be apa yang mudah untuk dilakukan oleh patient thalassemia because cannot be a very strong exercise. Maksudnya exercise yang sangat kuat. No, it's just minimal, okay. Tapi dia boleh dilakukan berulang kali. That's a very good things lah to help the thalassemia patients. To improve dia punya improve dia punya backbone problems. Okay.
Okay. Thank you, Dr. Isma and Mariana, for the questions. Next question. So still for Dr. Isma from Grand Kansa Valerina, Stikes Wirahusada, Jakarta. Okay, so the question is, if thalassemia suffers to some surgery, such as splenectomy or spinal cord transplantation, can they be declared completely cured of thalassemia?
Okay. Okay, thank you for the question, Grand Kansa Valerina. So untuk for the questions, okay, this patient who are removal of the spleen already, called the splenectomy, okay, they are not free of the thalassemics, but with reductions to their bodies. That's why we remove the spleen to prevent the infections very serious to the patients. So the way of the treatment, one treatment we try to suggest is, yes, of course, we have the bone marrow transplants, but the successful is depend on the situation of the patient. For example, if the patient between the bone marrow transplant happen between the siblings is more than 90%. So if the between the children patient and fathers or mothers, okay, the chances is around 60 to 80%. We have the patient that before do the bone marrow transplants, but it's very unlucky after the 6 months do bone marrow transplants, that kind of the kids too are going to be dead because the seriousness complication after the 6 months. That's the point of the things.
Thank you, Grand Kansa. Next question. So still for Dr. Isma from Angeline M. J. Gaud from Stikes Gunabasa, Jakarta. In some countries, gene therapy for people with thalassemia has been available to help with the treatment process. What is the potential of this gene therapy in the long term, and how will it impact the quality of life of the patients?
Okay. Yeah. So we have the another one alternative for called the gene therapy. But a few years back, okay, kita baru the one of the so many kind of the complications after they did the gene therapy. One of the things here adalah have the some kind of the another complications is to happen to in the thalassemia patient. For example, HIV and HCV viruses is quite serious to the patient because during the gene therapy process, the medication that we use to is called from the viruses that from the HIV virus. So they'll be infected to HIV when it turns to the serang semula kepada patient. That times yang kita terpaksa berhentikan sementara waktu dan we try to improve back okay of the gene therapy procedure. Process.
All right, thank you, Dr. Isma. I think it's the last question for you today. Okay, next question. We have a question from Nadia Savwa, from Guna Bangsa to Grafinda Widyaswara. Are there any growth media that can be used to grow normal flora bacteria other than Na, sodium agar and MAC? And can NaMAC media be used as universal media for various types of microbes? Yeah.
Okay. Thank you, Mr. Wisnu. Thank you, Nadia, for the question. Yeah, there are any media that can used to be grow for normal flora bacteria, and such as PCA, yeah, and then BAP for blood component, especially, and then and can NaMAC media be as universal measures of microbes. For Na, yes, but for MSC, no. MKR is just for negative gram bacteria. Maybe that's all for
All right,
Nadia
next
question.
question, please. Okay, that's from Nadia Shafwa. And we have another question from Handoko, Stikes Guna Bangsa. The questions for Mrs. Grafinda. Does the use of NaMCA methods in the development and breeding of microorganisms have disadvantages and advantages?
Of course, metode NA, bukan metode ya, namanya nutrien agar dan MCA media ya, bukan metode, memiliki kekurangan dan kelebihan pasti gitu. I will answer in bahasa, is it okay, Mr. Wisnu?
Yes.
Oke, NA media memiliki kelebihan dia bisa menjadi media bagi semua jenis bakteri, sedangkan untuk MCA media hanya terkhusus bagi pertumbuhan gram negatif. Maka tadi di penelitian juga dijelaskan bahwa yang tumbuh atau yang adalah bakteri gram negatif. Sedangkan untuk kekurangan dari NA sendiri, karena semua bakteri bisa tumbuh, sehingga kita tidak bisa concern kemana jenis bakteri yang ingin kita dapatkan. Seperti itu.
Okay, it's really clear. Thank you, Ms. Grafinda. Another
Okay.
question? We have more questions here.
Oh, for me. Okay.
Stephanie Latuasen from Guna Bangsa. Apakah di PMI Sleman terdapat UV sterilisasi pada setiap ruangan? Jika iya, bagaimana mungkin terdapat pertumbuhan bakteri di ruang AFTAP maupun IMLDD PMI Kabupaten Sleman? Apakah penggunaan UV sterilisasi belum dapat menghambat pertumbuhan bakteri?
Oke, di PMI Sleman khususnya di ruang IMLTD atau TTI tadi sudah ada UV sterilisasi, namun memang penggunaannya baru setelah dilakukan penelitian ini, begitu. Adapun untuk yang di ruang AFTAP sudah ada, mungkin di bagian atas begitu, tapi kurang maksimal sehingga sudah diperbaiki untuk proses CPOB yang tadi saya maksudkan gitu. Kemudian apakah penggunaan UV sterilisasi belum dapat menghambat pertumbuhan bakteri? Sejauh ini sudah, namun mungkin kita perlu waspadai dan perlu tingkatkan terkait dengan periode waktu dalam penggunaan sterilisasinya. Apakah 15 minutes itu enough, 10 minutes enough, or maybe 30 minutes enough begitu.
Alright, thank you, Stephanie. Next question. From Sekar Ayu, Politeknik Kesehatan Kemenkes Malang. Oh, this is for Dr. Fitri. What about the DNA found in the Treponema pallidum? Why this bacteria cannot be detected on N80? Is there a difference in the DNA titer of the bacteria so that it cannot be detected?
Okay, thank you for the question, Mbak Sekar Ayu. So I will deliver it in bahasa. Maybe bagaimana DNA yang ditemukan di Treponema pallidum, mengapa ini tidak dapat? Actually, I want to ask the specific what happened in maybe your research. Kenapa tidak dapat dideteksi di NAT ya? Mungkin bisa jadi gennya itu gen targetnya itu mungkin belum spesifik untuk Treponema pallidum sendiri. Ini kan yang menyebabkan sifilis ya. Or maybe there are prosedur yang mungkin dilakukan dalam NAT atau amplifikasi ini belum sepenuhnya benar ya, atau memang PCR ini adalah suatu tricky metode ya, jadi harus benar tentang penggunaan reagen atau ya volume dan sebagainya, suhu. Jadi seringkali researcher juga itu harus modifikasi dulu about the suhu, temperatur juga itu harus tepat juga ya, agar DNA target yang ada di Treponema pallidum itu bisa muncul di electrophoresis visualization. Kemudian apakah ada perbedaan titer DNA bakteri sehingga tidak dapat dideteksi? Kalau bicara tentang titer dan DNA itu sebenarnya beda kondisi ya, karena yang titer itu dengan pengencaran bertingkat mungkin ya, dengan using TPHA ya, itu untuk deteksi Treponema pallidum, tapi ini beda lagi dengan PCR itu harus pakai sampelnya itu DNA-nya, jadi harus melalui DNA isolation procedure dulu gitu. Mungkin semoga bisa menjawab untuk Mbak Sekar Ayu.
Yeah, I think it's really clear, Dr. Fitri.
Yeah.
Okay, do we have more question? Oh yeah, for Dr. Fitri from Variana Intansari, ABKPMI. Mungkin saya melewatkan beberapa presentasi, itulah topik yang menarik. Saya ingin bertanya tentang bakteri identifikasi yang berasal dari kantong darah atau isolasi. Mohon jelaskan. Ya. Dan kemudian bagaimana peneliti mendapatkan panen sel yang akan menjadi target gen.
Thank you, Variana ya. Identifikasi yang berasal dari kantong darah. Actually, we do not need. Jadi kita tidak butuh sampel darah yang banyak untuk identifikasi ini ya. Jadi mungkin beberapa mililiter dari sampel red blood atau mungkin blood products yang lainnya untuk dilakukan amplifikasi ini. Bisa juga kita melakukan gold standarnya yaitu melalui culture. Jadi sampel itu kita tanam dalam media atau kultur ya di laboratorium untuk melihat bakteri apa saja yang bisa tumbuh pada media tersebut. Jadi bisa dengan dua cara sebenarnya, yaitu ya dengan culture maupun dengan molekular method yang tadi ya. Jadi bisa memilih. Jadi masing-masing ada kelebihan dan kekurangannya. Kemudian bagaimana peneliti mendapatkan panen sel yang akan menjadi target gene. Nah, kalau panen sel sendiri, harvesting cell, we have to grow the bacterial from the sample. Jadi kita lakukan culture dulu, kemudian kita bisa memanen bakterinya, kemudian kita tampung ya dalam suatu mungkin buffer atau bisa juga pakai NaCl, saline water, kemudian kita putar ya, kita pusingkan menggunakan centrifuge. Jadi nanti kalau setelah kita pusingkan, jadi kan pusing gitu, jadi sel-sel bakterinya itu kan kayak komedi diputar gitu ya, jadi saling bertabrakan ke dinding-dinding dari tube-nya itu. Nah, nanti kita akan dapatkan peletnya. Nah, itu yang kita gunakan untuk keperluan selanjutnya, misalnya untuk PCR method tadi. Seperti itu. Mungkin semoga menjawab.
All right, yeah. It's a tough question, I think. Yeah, thank you, Dr. Fitri. Okay, from Aryo Sanjaya, Stikers Guna Bangsa, for Dr. Fitri, can DNA isolation be done on all living things other than viruses or bacteria, and give the reasons and what factors influence the success of DNA isolation?
Oke ya, principally secara prinsip, jadi we can do isolation DNA, melakukan isolasi DNA itu pada semua makhluk hidup yang punya genetik material genetik, baik itu DNA atau RNA. Jadi RNA itu biasanya ada di virus ya. Banyak virus itu tipenya adalah RNA virus. Asalkan kita tahu prosedurnya bagaimana dan itu sudah banyak kit ya, komersial kit already in market, jadi kita bisa pesan kit yang sesuai dengan apa yang ingin kita lakukan. Misalnya isolasi DNA itu sudah ada kitnya dan yang traditional method itu juga sudah ada. Ataupun dengan sampel yang dari virus tadi itu pun sangat bisa kita lakukan ya dengan prosedur yang benar. Jadi baik manusia tentu saja bisa ya. Dulu saya penelitiannya menggunakan urin. Jadi urinnya itu diduga mengandung mikrobiom atau bakteri-bakteri yang banyak sekali. So we have to do the isolation DNA first, isolasi DNA. Kemudian baru kita dapatkan DNA peletnya tadi ya, kayak endapannya itu. Nah, kita gunakan untuk step selanjutnya. Nah, itu tinggal ikuti saja prosedurnya, kita nanti dapat mendapatkan sampel DNA from the sample ya, entah apapun itu. Cuman memang itu need pengalaman ya atau experience. Jadi sometimes kadang the sample is very low. Jadi bagaimana kita mengatasi permasalahan misalnya sampelnya itu kemungkinan DNA-nya itu sedikit sekali. Nah, itu kita bisa melakukan sejumlah trik. Misalnya kita perkaya dulu misalnya dalam suatu nutrien broad media, jadi itu bisa agar bisa tumbuh banyak bakterinya. Setelah itu baru kita harvest ya atau kita panen. Kemudian kita lakukan tahap selanjutnya misalnya purifikasi, PCR, kemudian elektroforesis. Kira-kira seperti itu. Thank you for the question, Mas Aryo.
Oh, Aryo Sanjaya. It's a difficult question, I think, right? Okay. All right, everyone, thank you so much for actively participating and joining the research sharing with our incredible speakers. So we come to the end of the session. Once again, thank you so much, Dr. Isma, Mrs. Gravinda, Dr. Fitri, for sharing your research. And to give and to show our gratitude, our appreciation, we will give the certificate of appreciation virtually here. So the first one, certificate for Dr. Isma. So this is from the from our chief and our head program head, Mrs. Wiwit. And then the next speaker, Mrs. Gravinda, we will also give the certificate of appreciation to Mrs. Gravinda. Or no need? Nah, nggak perlu ya? We have, right? Okay. So for Mrs. Gravinda first. So thank you, Miss Gravinda. And the last one for Dr. Fitri. Thank you so much for your research sharing. All right. Okay, thank you so much once again for joining and participating at this session. And this is a really good opportunity for us to exchange the idea, exchange the experiences, the practices, and the research from the Malaysian perspective, from the Indonesian, from blood bank, from medical laboratory. So I do hope that this session will give you a lot of learning and lesson. So it's enough from me, and I'm singing off now, and over to you, our MC Anissa. Thank you.
Okay. All right, everyone. That's an amazing session with our incredible speakers. Thank you, Mr. Wisnu, for moderating this session. So we have an interactive discussion. Thank you to all participants for the incredible enthusiasm during the Q&A session of our international webinar today. From the many great questions, we have selected 3 best questioners who will receive special prizes. Okay, the best questioners are the first is Nadia Safwa from Stikas Guna Bangsa Yogyakarta. Okay, the second is Mariana Placidia Bali from Physiotherapy Stikas Guna Bangsa Yogyakarta. And the third is Variana Intansari from ABK PMI. Congratulations to the top questioners. Please feel free to contact the committee through the provided contact details to claim your prize. Thank you again for your participation and keep up the great spirit of learning together. The upcoming session is what we are waiting for, especially for the poster competition participants. Since we will be announcing the winners, we are so thrilled to receive so many posters. It was not easy to decide because all of the posters are amazing. All right then, get ready and take a deep breath. The second favorite poster is congratulations to Intan Miftah Nur Aziza from Politeknik Kesehatan Kemenkes Malang. Okay, next, the first favorite poster is congratulations to Mikael Emma from Universitas Dr. Sutomo. Okay, next, the third winner is yay! Congratulations to Edwin Yeremia from Stikas Guna Bangsa Yogyakarta. Okay, next, the second winner is congratulations to Winda Septiana from Stikas Guna Bangsa Yogyakarta. And our first winner goes to Samtung Guna Bangsa Yogyakarta. Okay, congratulations, guys. Once again, congratulations to all our winners. For those who didn't make it, no worries. Keep trying, keep fighting. we might have other competitions in the near future. Thank you much. Thank you so much for submitting your wonderful artwork. All right, everyone. We have come to the end of our webinar. We really appreciate everyone who support this event. Special thanks to Dr. Isma and the student from University Technology Mara who flew away from Malaysia to exchange knowledge and experience with us. Once again, thank you so much everyone for joining and actively participating in this webinar. I'm Anissa Handayani. See you at our next event. Wassalamualaikum warahmatullahi wabarakatuh. Alright everyone, we will have another event in this afternoon, a guest lecture from Akita University, Japan, Professor William Bradley Horton, who will give us a lecture about public health and malaria from Dutch colonialism and Japanese occupation.